remember to turn on the laser pointer at this time. There we go. Okay, so our next speaker is Jenna Kuhn from the lab of Dr. D in the Department of Environmental and Occupational Health to speak about antimicrobial resistance development timing of MDR, Klebsiella pneumonia to last resort antibiotic colistin, which I'll tell you, this is something that we see clinically and it's bad. No explaino, big problem.
Good afternoon. So as we all know, the threat of antibiotic resistance is global and continuously getting more and more challenging to treat. So in 2019, the CDC has actually developed this priority list of critical pathogens. And these are to focus in and hone on which ones are most important for drug development. So Klebsiella actually falls under this carbapenem-resistant Enterobacteriaceae, and this is considered a pathogen of urgent concern. So there's actually two pathotypes of Klebsiella that evolved independently. So we have classical KP and we have hypervirulent KP. Classical KP is mostly associated with nosocomial, so your hospital-acquired infections, and a lot of these strains have acquired antibiotic resistance factors, so like genes that encode for enzymes that actually degrade these antibiotics. Hypervirulent KP are associated with community-acquired, so they can infect otherwise healthy individuals. And these strains were previously susceptible to most antibiotics and obviously contain virulence factors that make the infection more severe. But now, as we all know, we have this convergence of both antibiotic resistance and hypervirulence, creating this sort of monster superbug. So we have carbapenem-resistant hypervirulent Klebsiella. Just last month, the World Health Organization published this priority list of drugs for use in treating children and neonates. So essentially, they're working to design drugs that have higher efficacy, safety, and toxicity. It's another priority, and they're also designing these drugs to be more efficient and effective for these more complicated resistance patterns we're seeing in bacteria. And from about 2012 to 2017, we've seen relatively stable levels of creatinine, and this is with the continued action. And unfortunately, we've seen 50% increase in extended spectrum beta-lactamase producing Enterobacteriaceae. So according to this optimization, they have phase 1 compounds in development, and essentially they're targeting the multidrug-resistant Klebsiella. And they're also using what's called a carbapenem sparing regimen. So the less we're using these important antibiotics, hopefully there's less development of resistance. We're reserving them as last resort treatments. So to really hone in on the severity of the situation, we have here two clinical isolates. We have 107 and 105.2. These are both Klebsiella isolates taken from human patients. We have a panel of many antibiotics. There's at least 5 different classes there. And colistin is one of the only ones that really shows effective ability to kill these superbugs. We also have a synthetic peptide known as v345 we're working on in our lab under clinical development that is also able to kill these organisms. So like I said, colistin is reserved for last resort in the most severe multi-drug-resistant infections. And in a few retrospective studies, it has shown high efficacy and low levels of nephrotoxicity in neonates and young children, and is also often used as a combined therapy. So in really trying to study development of colistin resistance outside of the human, essentially we're trying to just mimic what's going on in a test tube in vitro. To do this, we account for both bacterial lifestyles. So I do a daily planktonic transfer into or a daily biofilm colonized bead. The bacteria grows on the bead, and we transfer a single bead into media and supplement this media with increasing concentrations of colistin. And from here, we do what's called minimum inhibitory concentration testing to really be able to visualize the increase in MIC over time. So this means minimum level of drug required to inhibit the bacterial growth. And finally, based on the MIC result, I selected populations of interest to do whole genome sequencing and mutation prediction analysis. So I'm looking at the chromosomal aberrations contributing to colistin resistance. So this shows a 9-day in vitro colistin selection experiment using hypervirulent KP strain 43816 in blue in 2 clinical isolates, 107 and 1052, which acquire rapid colistin resistance, quite obviously. So you see the baseline level between 1 and 2 micrograms per mil, and everything in this gray background is considered above the clinical breakpoint. So not even a viable treatment option at that point. And we see upwards of 256 fold change increase in MIC just within 9 days of treatment, and just exponentially increasing for isolates 107, greater than a 16,000, I'm sorry, fold change increase in MIC. That just goes to show how substantial the resistance is. I can't even recognize a number. But another thing that we're interested in is why are there different trajectories in the resistance pattern here? We don't see them all evolving at a similar rate or a similar pattern. And another thing that's interesting is, number one, between day 0 and 1, we have this extremely high rate of resistance. And this is treating at 1/2 level MIC, a double treatment after every 3 days. And by the end of the experiment, we see, isolate 107 reach much higher and possibly even further increases in MIC, where the other 2 isolates actually kind of level out. So basically, I wanted to magnify in on what kinds of mutations are occurring in this hypervirulent KP. So I took both planktonic and biofilm cultures and did the whole population sequencing to really identify the starting point for various mutations and see how long they will persist in each evolving population. So if you can see the MIC background, you see sort of a pattern of this increase along with a new generation of mutation. And these mutations are occurring and progress in sort of like association with the resistance progression. So we're trying to understand the importance of the timing of these mutations and which ones might be critical for the generation of further mutations. So we did see a pattern. We saw mutations in virulence factor RNPA occur within 1 day of treatment, and then we saw mutations in LPS modification and LPS biosynthesis. So RNPA is probably leading to a loss of capsule polysaccharide production. And next, PHO-Q and PMRB are involved in modification of LPS. So they're involved in forming these compounds that are or molecules binding to the lipid A of LPS, and therefore colistin is unable to bind. So that's a resistance mechanism, and also LPS biosynthesis and cell membrane integrity, meaning efflux. So we designed, based on all this information, we developed these timelines in both planktonic and biofilm lifestyle, and actually found very similar results in terms of the mutation timing between lifestyles. So in summary, we hypothesized that RNPA is a critical initiating mutation that is basically making the bacteria a little bit more vulnerable. At the same time, it could be a trade-off between virulence and increased antimicrobial resistance. So the next, we see some efflux and regulation in osmotic pressure and energy metabolism. And again, these modifications of LPS and LPS biosynthesis. So as you can tell, the main targets of colistin is the cell membrane. So colistin will bind to this lipid A, which is embedded in the membrane, and this basically offsets the charge of the membrane, leading to membrane perturbation and expulsion of the cell's contents. So luckily, there is some positivity in drug development because we are designing these dual antibiotics that have different classes of antibiotics to hopefully prevent or lessen resistance. So if you remember the huge fold change increase in the strain 43816, this shows the same strain but tested against ceftazidime avibactam. So ceftazidime avibactam is a third generation cephalosporin combined with a beta-lactamase inhibitor. So basically the beta-lactamase inhibitor prevents that beta-lactamase enzyme from inhibiting ceftazidime activity, making it resusceptible to treatment. So we do see this strain is susceptible to this treatment, although it is extremely colistin resistant. And what I was mentioning before is what we see as a potential trade-off between increased antibiotic resistance and hypervirulence. We see less of a mucoid phenotype, which is one of the potentially less accurate, but one of the key features of hypervirulent KP. So my current kind of approach is using targeted gene knockouts. So we know important gene functions or cellular functions involved in the resistance mechanism. So we can target different genes that have mutations occurring at different times throughout the resistance progression. We can study individual gene knockouts, 2 combination gene knockouts, and 3 combination, and see the impact of antimicrobial resistance. Is there a synergistic effect? Is there an additive, or is there no effect? So in conclusion, mutations in the gene RNPA may be critical for initiating this resistance in hypervirulent KP. There could be an evolutionary trade-off between increased antimicrobial resistance and reduced hypervirulence. We can see mutations occurring sequentially or simultaneously, meaning multiple mutations all at one time. And we hypothesize that timing and synergistic effects of resistance mutations may largely impact the development and progression of colistin resistance. And we do think and have seen that ceftazidime avibactam is a critical therapy for colistin-resistant infections. In the last minute, I will talk about future directions. So currently, I'm studying mechanisms of KB resistance to ceftazidime avibactam using CRISPR-based gene editing. And I would like to take these mutants and study how their mutations lead to differences in their virulence in a mouse model. And we're also trying to discover or learn more about the clinical potential for novel AMPs. So like I mentioned previously, our lab is focusing on a novel synthetic antimicrobial peptide V345, which has shown significant inhibition of biofilm growth and a few species of escape pathogens. And all this information could be really interesting and important in possibly designing a sort of computational method of predicting resistance. So if we have the whole genome sequence of isolates we obtain from patients in the hospital, can we do a quick screening of some sort to detect the potential for certain mutations, which then we link to the actual drug that sort of selects for those mutations because of the pressure, the environmental pressure. So with that, thank you for allowing me to share our research, and I'll take any questions.
Thank you. Really interesting and important stuff. We see patients with these infections in the hospital all the time. I'm curious if you can comment on the fitness costs. You talked a little bit about loss of virulence in this in vitro assay, but the fitness cost to the bacteria in the development of colistin resistance. And then kind of a secondary question to that would be, how does that affect the ability of that bacteria to compete in the big microbiome of the host?
Well, I don't think we've actually done like a fitness assay, but I assume that changes in virulence would be linked to fitness. So I'm not sure how well we can actually study that accurately in vitro. I think it would be really important to study that in a mouse model of infection. So that could be definitely an interesting thing for me to study in the future. Can you repeat the second part?
The second question was, K pneumo and all these other Enterobacteriaceae, they exist in a big microbiome in our bodies. And so if there's a fitness cost to the K pneumo to have colistin resistance, would that be lost in the in vivo setting, right, of the microbiome?
I would say that's a very complicated thing to actually study. Because there's obviously got to be a balance of good versus the bad bacteria. And I'm not really sure. I'm not really sure if I can have a good answer for that, but that is a very good question. Thank you.
I mean, certainly, there's lots of folks here at the institution who study the microbiome.
Mm-hmm.
I'd be interested in helping with that work.
Mm-hmm. Thank you.
I had a comment to the question. It's really interesting, the trade-off you mentioned between resistance and fitness, because we see that in other microbes. As Anna mentioned earlier, we've been involved with the Pittsburgh phage project, and often with phage therapy for MDR bacteria, there's a trade-off where as the bacterium becomes resistant to phage, it then becomes susceptible to antibiotics again. And we now have a lot better HIV treatments, but we used to see as patients with HIV developed, as their virus developed resistance, the virus became a little bit less fit. So it's just interesting that you see that here too. I wondered, it's a little scary how quickly they all develop resistance, in this model. It looked like the strains all got to the same place, basically, but they started with different mutations. So is that, do you think, a feature of the strain that it sort of chose that mutation first, or is it just chance that a mutation in that gene occurred that then gave it a selected advantage for that pathway, that metabolic pathway?
Right, and we're certainly trying to better understand how the stability of the genome can affect, the possibility of this strain just randomly acquiring a beneficial mutation. And with clinical isolates, they have a lot more, they're more likely to have acquired certain resistance genes through the ability, because they're in the body, they're surrounded by different strains, and it's a whole microbiome versus in a test tube, you have one population evolving. So you have the shared antimicrobial resistance factors just being shared between organisms. So we can't actually say that, based on the certain genome of the clinical isolate, that it's definitely going to acquire a certain mutation. It definitely depends on the environment. But I think it's the most important thing is the drug under selection, because that's a certain target on the bacterial cell. So whatever you're exposing the bacteria to, unless it has innate resistance, is probably going to develop resistance to that drug, depending on which genes are most important in that. So depends on basically the background of that strain, how it's evolved in humans, and then what kind of selection pressure it's been in just by the patient being treated to multiple drugs, and the severity of that infection is a big thing. It's a complicated, hard thing to study.
This is why I'm a virologist.
Sorry, it's not. I don't want to have all the ID doctors come up and ask you questions, but I was very interested and frankly horrified, like John, about the rapid emergence of the colistin resistance. But then later, and I've thought of this separately, you showed, I know Tom Hooven and other folks have done work with CRISPR to sort of change the gene expression of the bacteria.
Mhm.
And it made me wonder if you've ever, if you've explored CRISPR to change like reversion of colistin resistance or any antimicrobial resistance. And by extension, this is outside my field of study, but we're doing, or not we, writ large, folks are doing lots of CRISPR-Cas delivery in variety of clinical and in vivo settings. And I don't know if you could maybe comment on where that stands in the field of antimicrobial resistance.
Right. It's definitely developing more and more because a lot of CRISPR now is focusing mostly on mammalian cells and diseases, but there's been a lot more focus, I guess, more recently on being able to generate gene knockout or insertion, making specific modifications using different plasmids. But I would say it's, you have to know the function, whether it's gain or loss of function mutation is probably the most important thing before you even design this type of like gene mutation that you're trying to do. And a lot of it helps whenever you actually do the whole genome sequencing and do the mutation prediction. You can see where the mutation actually was generated in the whole genome. And then from there, you can maybe target that area for either if you're trying to create loss of function, then you can knock it out, or you can restore that function by creating a knock-in and see its impact. And there's even more accurate ways now of just using certain CRISPR models where you can just do a certain single base pair modification. So you can really have these intricate designs based on what kind of mutation you're trying to develop. But as a warning, I think the most difficult thing is designing the appropriate target. So you have a spacer that targets the Cas9 to the correct area. And we found that it's mostly that specificity and the potential for off-target cutting has been the biggest challenge probably. And in bacteria, we have a lot less reference genomes to use for these softwares that detect this off-targeting. So not to complain, but that's a hard thing to manage when you don't have, a reference to use. So. Thank you.